For this experiment, I will be examining how sugars are in both smoothies and your body. My question states, how do people examine sugars in smoothies and determine how enzymes convert them into energy? My hypothesis says if one changes the type of sugar and enzyme that are dissolved in a solution, the percentage of glucose concentration will vary. The independent variables are the sugars and enzymes, while the dependent variable is the percentage of glucose. Some materials needed are glucose helpless, invertase, a blender, and any other materials. The procedure starts with a control test to make sure the test strips are working, which is my control group. Next, invertase and lactase enzymes are tested in a sucrose and lactose solution to measure conversion speed. Finally, food samples undergo a test to identify which sugars are in each food sample. These last two steps are my experimental group. As you can see, the projected and actual results in this control test are the same. Since they match, the test strips can be used. Next, by mixing water and sucrose and water and lactose, a 10% sucrose and 10% lactose solution is formed. After adding invertase to sucrose and lactase to lactose, measurements are held every 5 minutes during the first 30 minutes and held every 10 minutes until 90 minutes have passed. In this final test, additives are diluted and both the liquid and additive food samples are measured in a 3-part data table consisting of one without enzymes, one with invertase, and one with lactase. The two graphs on the left represent invertase and lactase, which converts sucrose and lactose. On the right, the liquid samples are on top and the additive food samples are on the bottom. As you can see, all the graphs are different. To conclude, my hypothesis was fulfilled. For example, in this digestive enzyme chart, the majority of the glucose percentages change depending on the two independent variables. In human bodies, low sucrose levels represent that their invertase enzyme is most likely impaired. Likewise, if they have low lactose levels, the lactase enzyme is most likely messed up, which is also known as lactose intolerant. From the food samples shown, one can see that when a sample's glucose percentage increases, the sample is matched up with a certain enzyme. For example, milk rises at lactase to 2.5%, and apple juice rises at invertase from 0.5% to 2.5%. This means milk contains lactose and apple juice contains sucrose. Diabetics and other victims can use this knowledge because it is cheap and simple to follow through with. It gives victims an idea of a healthy diet which improves both themselves and society. Thank you. #Google Science Fair.